Hi everybody, and welcome to our module on genetic mapping. Let's start by reviewing the principle of genetic recombination. During meiosis, chromosomes exchange segments so that the child inherits a patchwork of parental chromosomes and never an exact copy of one of the parental chromosomes. To understand this, let's imagine we have a father and mother with two copies of a chromosome. For example, let's assume this is chromosome number 15 here, and the father has two copies and the mother has two copies. Those chromosomes are going to recombine during meiosis such that child 1 inherits one copy of chromosome 15 from his father and one from his mother, but that chromosome 15 from the father is a mixture of the two chromosome 15s that were present in the father's cells, and the same is true of the mother, and this mixing of genes is called genetic recombination. The next concept we need to explore is what's known as independent assortment. To understand this concept, let's suppose a father has two alleles of his F gene and two alleles of his M gene. Let's suppose that one of his F genes is in a form that we'll call capital F, and the other F gene has a different form which we'll denote as lowercase f. Let's also suppose that he has two versions of the M gene, one is capital M and one is lowercase m. Let's further suppose that F and M are found on different chromosomes. If this is the case, the F and M genes will independently assort. This means that they can independently recombine, and we'll get a 25% chance of each mixture of the F and M genes in a gamete. To make this more clear, let's look at this picture I've drawn on the screen. So we have a father who has chromosomes within the cells of his body. On chromosome 1, he has the F gene, and one of his copies of chromosome 1 has the capital F gene. The other copy of chromosome 1 contains the lowercase f version of the F gene. Similarly, on the father's chromosome 2, he has two copies of the M gene. One is capital M, the other is lowercase m. Because these genes are found on different chromosomes, they will independently assort, and the gametes will be a mixture of all the possibilities. In other words, 25% of the gametes will contain a capital F and a capital M gene, 25% will contain a lowercase f and a lowercase m gene, and then 25% will contain mixtures of a capital and lowercase f or m. Now in that example, the F and M genes were able to independently assort because they were found on different chromosomes. But now let's consider what would happen if the F and M genes were found on the same chromosome. To understand this, let's look at the example I've made on the screen here. Let's assume we have a parent with two copies of chromosome 1 found in the cells of their body. Each copy of chromosome 1 has an F and an M gene. On one version of chromosome 1, there is a capital F and capital M gene. On the other version of chromosome 1, there is a lowercase f and lowercase m gene. What would happen if this parent were to generate gametes? What combinations of the alleles of the F and M gene would they have? Well, it turns out if the F and M genes are very far apart on chromosome 1, then crossover will occur in meiosis to such a degree that you will get the same mixture of combinations of the F and M alleles as if the F and M genes were found on separate chromosomes. In other words, there's a 25% chance that you would create a gamete with a capital F, capital M version of the F and M genes, a 25% chance that you would create a gamete with a capital F lowercase m, and also a 25% chance of the two other combinations. And this is what occurs if the F and M genes are very far apart on chromosome 1. In this situation, we say that there is a 50% chance that the gametes will have a parental version of the F and M genes. In other words, there's a 50% chance that a gamete will have either a capital F, capital M gene, just like we found in the parent, or a lowercase f, lowercase m gene, just like we found in the parent. There is also a 50% chance that the gametes will have what's known as recombinant versions of the F and M gene. In other words, a capital F, lowercase m, this is a mixture of F and M genes that was not found in the parent, or a lowercase f, capital M, another mixture not found in the parent. Now let's consider what would happen if the alleles of the two genes are very close together. In this case, there is very little crossover, and there will be a low occurrence of the recombination genes, capital F, lowercase m, or lowercase f, capital M. At the extreme, you would get what I've shown on the screen here. If F and M genes are very, very close together on chromosome 1, you would get 50% of gametes having a capital F and a capital M, just like you had in the parent, a 50% chance of having a little f, little m, just like you had in the parent, and you would have a 0% chance of getting any of the recombinant chromosomes because no recombination is occurring because the genes are so close together on chromosome 1. The reason there is little crossover and recombination when genes are close together is because of what I've shown on the screen here. Imagine that the A and B genes are very close together. Only a break in this small section of DNA is going to allow them to recombine. Now imagine that the B and C genes are very far apart. There is a huge span of DNA in which any break will allow B and C to recombine. Thus, you are going to get lots more recombination of the B and C genes relative to the amount of recombination you will get of the A and B genes. So now we can define an important term known as recombination frequency. This is the frequency of recombined genes. In the example we went through on the last few slides, this would be gametes with a capital F lowercase m or a little f capital M. Recombination frequency is denoted by the Greek letter theta, and it ranges from 0 to 0.5. If we go back to this slide here, in this example, genes F and M were very far apart, and the frequency of recombined gametes was 50%. This would be a theta of 0.5. And on this slide, where we considered what would happen if the F and M genes were close together on the same chromosome, there was no occurrence of the recombined genes in the gametes, and therefore this would be an example of theta equal to 0. And a key point here is that the recombination frequency is proportional to the distance between the two genes. If the genes are close together, theta will be 0. If they are far apart, theta will be 0.5. And this can be used to create a genetic map of our genes. So now we've gone through all the concepts that we need to understand in order to discuss genetic mapping, which is also called linkage mapping. This is done by studying large families and tracking the frequency of genetic recombination and using that frequency to determine relative gene locations. So for example, let's suppose we have 3 genes, A, B, and C. Let's suppose that the frequency of recombination of A and B is 16% in a particular family. Recombination of A and C is 0.08 or 8%, and recombination of C and B is also 0.08 or 8%. This means that A, C, and B must have relative locations on a chromosome like I've shown at the bottom of the screen here. A and B are so far apart that they generate a recombination frequency that's higher of 0.16. A and B are far apart such that they generate a recombination frequency that is smaller of 0.08. This is called linkage mapping or genetic mapping, and it's used to map our genes. This brings up an important term that we need to discuss, and that is the term linkage. This is the tendency of genes or alleles to transmit together. When alleles are more linked, they display less independent assortment. Genes that are close together that have a theta closer to 0 are said to be tightly linked. Genes that are far apart and have a theta closer to 0.5 are said to be unlinked. I'm going to finish this module by discussing the concept of linkage disequilibrium. This is used to study genes that are very close together. When genes are very close together, recombination events are rare and family studies are impractical. Even if you have a large family, you may never see a recombination event, and therefore you'll never be able to calculate a value for theta. Therefore, the principle of linkage disequilibrium is used to study these types of genes, and this is done by studying large populations, not families. Instead, this is applied to populations of individuals. To understand linkage disequilibrium, let's first talk about linkage equilibrium. And to understand this concept, let's assume there are 2 genes found in a population of individuals. Let's assume gene A exists in 2 polymorphisms. There is a big A allele and a little a allele. Let's assume that big A is found in 50% of individuals in a given population, and little a is found in the other 50%. Let's also assume that gene B has 2 polymorphisms. It can exist as a capital B or lower B allele. Let's assume big B is found in 90% of individuals and little b in the other 10%. And I've listed these numbers at the top of the screen here to remind you what they are. Once we know the percentage of individuals with each of the different alleles of the A and B genes, we can then determine what the population frequencies should be for mixtures of those different alleles. The frequency of a big A and a big B found together should be 0.5 times 0.9, which is 0.45. Similarly, a little a and a big B together should be 0.5 times 0.9, which is 0.45. And we can go ahead and calculate all the expected frequencies of the mixtures of those alleles. And if this were to be what we actually found in the population, we would say that these 2 genes demonstrate what's known as linkage equilibrium. And just a side note here, don't think that you can apply the Hardy-Weinberg law to these gene frequencies here. The Hardy-Weinberg law is meant to determine the frequency of the same gene with different alleles occurring together. In other words, the frequencies of big A's and little a's occurring together. Here we're talking about 2 different genes, and in this situation, the Hardy-Weinberg law does not apply. So now that we understand linkage equilibrium, we can talk about linkage disequilibrium. This occurs when the population frequencies are higher or lower than expected. For example, imagine if we found an actual mixture of the big A and big B genes in a population of 0.75. That is much higher than the expected frequency of 0.45. This means that this haplotype AB is in linkage disequilibrium, and genes will display greater degrees of linkage disequilibrium when they are closer together, and thus you can use the degree of linkage disequilibrium in a population to study genes that are very close together, too close together to study using families. To understand this more, let's consider a new gene mutation that we'll denote as capital A, and let's suppose that this gene mutation occurs in a gene that is very close to gene B. Initially, A and B are going to be transmitted together in a population. They are so close together that they will be transmitted together from one generation to the next. Now, over time, A and B may eventually separate. Random chance may eventually create a break just between A and B so that they separate. The likelihood of these two genes separating depends on their distance apart from one another and the size of the population. In other words, how many generations have had the opportunity to split A and B from one another. The degree of linkage disequilibrium will be greatest when a gene first enters a population, in other words, when the mutation first occurs. In that situation, A and B are going to be right next to each other, and you're not going to find them separated from one another on many occasions. With successive generations, the degree of linkage disequilibrium will fade. In other words, as the population grows, you will see less linkage disequilibrium. It will also fade if the distance between A and B is greater. So I've summarized these points on this slide. Linkage disequilibrium is affected by the genetic distance between two genes and the times the alleles have been present in a population. And a key point here is that different populations can display different degrees of linkage disequilibrium. If a gene has moved through a population of people for a longer period of time, in other words, if that was the population that first developed a mutation, they may display a different degree of linkage disequilibrium compared to another population that developed the mutation at a later point in evolution. And that concludes our module on gene mapping.